All right, good afternoon. Let's get started. All right, so my husband and I were talking at dinner time last week, and the Entner-Dodorov pathway came up, as it does sometimes at dinner. And so the question that we had last time is, how does the cell know that it's missing this enzyme? The question is, it probably doesn't. So, or the answer is it probably doesn't. So this enzyme that catalyzes this reaction from fructose 6-phosphate to fructose 1,6 biphosphate, or I think biphosphate is the way we say it now. That reaction is actually reversible. Not all of the reactions are. So like this step, the splitting in glycolysis, that is not reversible. Once that molecule is split, it can't be put back together again. But this is reversible as some of the other ones are. So, and plus this molecule, fructose 6-phosphate, is used elsewhere in the cell. So if it kind of heads down that pathway and then has to back up because there's a problem and it has to stop, it doesn't have this enzyme, this enzyme right here. It's not a big deal. So hopefully that helps answer the question that was asked in class on Wednesday. And then I wanted to start with just a little review, a little practice question here about the Entner-Dodorov pathway. So take a second, probably some of you have been looking at it as you've been waiting for class to start. Take a second, read over, make your best guess. I'm hearing some whispers. I'm hearing some C whispers. Yep. So that's correct. The other name we had for glycolysis, remember, it was Embden-Meyerhoff harness, or EMP pathway. Glycolysis is common to most forms of life, but remember Entner-Dodorov, there's only a few bacteria that can do that. It is a catabolic pathway, not anabolic, and usually an organism would do either glycolysis or Entner-Dodorov. It's not going to do them both at the same time. It will do pentose phosphate at the same time as either Entner-Dodorov or glycolysis, but not Entner-Dodorov. So, okay. Questions as we move on through respiration. All right, so where we left off last time, we just had gone through that transition step between glycolysis and the Krebs cycle, where that enzyme A attaches to the acetate. We said that only 2 carbon compounds will fit into the Krebs cycle. So that's what we find here too. Here, the acetyl-CoA combines with oxaloacetic acid to give us citric acid. All right, so I called this the Krebs cycle. This is named after Hans Krebs. He's the one that helped elucidate the pathway. We have several other names for this cycle. Anybody tell me one of them? Yeah, citric acid cycle, right? So named for one of the first intermediates there. So we also call this, again, citric acid cycle. What's another? There's one other famous common name for this. TCA, you probably heard that, tricarboxylic acid cycle. Oops, right in there, TCA. Okay, we call it that sometimes because there's lots of intermediates here that have 3 carboxylic acids, or at least several of them. Okay, so whether you call it TCA or citric acid cycle, Krebs cycle, that's all referring to the exact same thing. And this is taking place in the cytoplasm. That's for prokaryotes. For eukaryotes, it's taking place inside the matrix of the mitochondria. Okay, so mitochondria has actually two membranes, a regular plasma membrane, and then it's got this other membrane inside. So it's taking place inside that cristae, we call that other membrane. All right, so what's going on with this? First of all, if we think about the carbons, so we said we've got a 2-carbon compound combining with that 4-carbon oxaloacetic acid. Now we've got 6-carbon citric acid. But right away here, we've got a loss of a carbon by a decarboxylation as carbon dioxide here and here. So if you remember back in that transition step, we lost a carbon to decarboxylation there as well. So that 3-carbon pyruvate that we ended glycolysis with, we've lost all 3 of those carbons. So all 6 of the carbons in that original glucose are now gone to carbon dioxide. So that's kind of interesting to think about. There are some organisms that can't bear that loss of carbon, that they need that carbon to make structures. So there actually is kind of a modified Krebs cycle that some organisms will go through where they kind of shunt off and kind of skip losing that carbon. So that's kind of interesting. What else do we get from this? If you watch that Crash Course Chemistry YouTube video that I posted last time, he says it's all about the electrons. All right, we've got a lot of electrons coming out here, NADH, NADH, NADH here, as well as a different electron carrier that we only see in Krebs cycle, FADH2. So if you think about gains of the Krebs cycle here, we're getting usually, let's say, counting that transition step. Or if you remember, we gained an NADH from that. So we're going to get 4 NADH per pyruvate. You just have to be clear on how you're counting this. Are you counting it per pyruvate or per glucose? Are you counting it including that transition step or not? We're getting 4 NADH. And remember that NADH, that's carrying 2 electrons and a proton. Okay, so that's a hydride ion. So actually 2 electrons there. And we're gaining 1 FADH per pyruvate. Okay. What about ATP? You'll see that originally it's guanosine diphosphate that gets phosphorylated to guanosine triphosphate. That gets converted into ATP. So again, we're getting 1 ATP per pyruvate or 2 per glucose. All right, and how are we getting that? Remember, in glycolysis, we got that ATP by substrate level phosphorylation. Those phosphates were coming from some other substrate. Same thing here. We consider this substrate level phosphorylation as well. It's not as obvious where that phosphate is coming from. If you look at this succinyl-CoA, they don't draw any phosphate associated with that. I've been assured by a biochemist that the phosphate is associated maybe kind of transiently with that succinyl-CoA. So anyway, that's where the phosphate comes from. Another example of, again, substrate level phosphorylation. Don't need to memorize these intermediates. I'm more interested in what we start with. And here, the main things that are coming off of this. We could say, we could add 3 carbon dioxides per pyruvate. I'm not sure if that's anything that really helps the cell, but we could say, yeah, that's something else that's coming off of. So again, don't memorize those intermediates. I don't have those memorized. I'm not going to worry about enzymes or structures. What are we getting out of it? Okay, so the main thing we're getting out of it is reducing power, these electrons. Okay, so if we go back, I don't think oh yeah, I guess I did post this again. Go back to our overview of we're starting with respiration. So we're now ready to think about these electrons that are coming off of glycolysis. Where are they going? They are going to an electron transport chain. And that electron transport chain is always going to be in some membrane. Okay, for bacteria, it's in the plasma membrane. And it's really important that these sets of reactions are in an intact membrane, because just a little overview here before we get to a couple different slides to show you what's going on. It's going to be pumping protons outside of the cell. Okay, and that proton gradient, okay, anytime you got a whole bunch of something, it's trying to equalize out, it's going to actually diffuse back into the cell membrane without the cell spending any energy. So this is the water that's being held back by the dam, that when it does diffuse in, that powers the phosphorylation of lots of ADP to ATP. Okay, so we got that proton gradient that's being established. There's some electron acceptor. For us, it's always oxygen. Bacteria can often use something else, some inorganic ion, and we'll talk about what kind of things that could be. Okay, so that's just a little overview. Your book draws electron transport chains a couple of different ways. And one picture is a little bit better at showing certain things, and one picture is a little bit better at showing other things. Okay, so right off, notice this is the NADH, remember, that's carrying the 2 electrons and a proton, is getting oxidized back to NAD. Okay, so that's the oxidation. That's why any ATP that's gained here, it's a oxidative, it's by oxidative phosphorylation. Okay, it's not necessarily because oxygen is the final electron acceptor, but that NADH is getting oxidized. And you see how it's drawn in a diagonally. That's purposefully, because that's the energetically favorable way for the electrons to move. Okay, we can measure that. We can measure membrane potentials and actually measure that. I'm going to talk about that briefly in a little bit. But the electrons, think about them rolling downhill. And the electrons that are carried by NADH are kind of dropped off at the top of the hill. The ones carried by FADH2 are dropped off a little further down. Okay, so if you Olympics analogy, if you've been watching that, the men's losers, the top that start at the top of the hill, they get more speed up than the women's losers that have to start like 3/4 of the way up the hill. All right, so we get more ATP from the electrons that are brought there by the NADH rather than by FAD. They kind of enter a little different part of the pathway there. Okay, so what we have here is a series of electron carriers that are alternating between their oxidized and reduced states. Some of them are passing electrons and protons. Some of them are only passing electrons, and they're pumping the protons here. Like I wrote before, this is outside of the cell, and this is going to be inside of the cell. Okay, so they're pumping their protons out. And again, there's going to be an enzyme down here where the protons are passing through only at that one spot. If your membrane is not intact, if there's some holes in it and those protons are just diffusing in wherever they please, then ATP is not going to be generated. Okay, we only want them to come through that one enzyme, like the turbines in the dam, when the water passes through, that produces the ATP. What about these electron carriers? Don't memorize those because they are different depending on the organism. In general, what can we kind of say about them? You should sort of kind of recognize the main classes of molecules that serve as electron carriers. So down here, here's an example of our electron carriers. Some of them are flavoproteins, like this flavin mononucleotide, the first one here. What's a flavoprotein? It contains flavin, which is a coenzyme. There are several different kinds of cytochromes, and they're like cytochrome A, cytochrome C. There's several different kinds. Those are just proteins that have iron in them that alternate between oxidized and reduced state. There's a particular cytochrome that we can use in the lab that helps us identify organisms, because there's this whole group of gram negatives that don't have a particular cytochrome. We call them oxidase negative, and there's another big group of gram negatives that do, and we call them oxidase positive. So we can, again, that test can help us identify organisms. Ubiquitones, these are non-protein electron carriers. They're actually derived from one of the vitamins. Coenzyme Q is an example of an ubiquinone. And there's just some other molecules that are just made of heavy metals, like iron sulfide here. Again, important thing, it alternates between being oxidized and reduced, so it can pass their electrons down. Let's see, what did I forget to say? Okay, so let's look at the next graphic that your book shows. Again, it's nice because it reminds us where this is taking place. For bacteria, it's going to be in that plasma membrane. And over here for eukaryotes, it's going to be in the matrix membrane of the mitochondria. So that's that inner membrane of the mitochondria. That's where these carriers are going to be located. Okay, so the NADH oxidized to NAD, the electrons are passed, the protons are pumped out. Here, now this picture shows where the protons are diffusing back in. They're in an enzyme called ATP synthase. Okay, so our ATP is synthesized. It makes sense. I love it when names make sense. Okay, so it's here when the diffusion of those protons, all right, because we have a big proton gradient buildup, we call that a proton motive force. Okay, because there's so many protons there, they diffuse in, and that, again, it's kind of like turning the turbines and causes the phosphorylation of ATP to ADP to ATP. Okay, and we call that process semiosmosis. Okay, so that's the movement of, I'm running out of room here, I'll write it up the side. Semiosmosis. Okay, the diffusion of these protons back into the cell. Okay, let me come back to this in a second, but I found another slide that kind of explains that process maybe a little bit better, because those two terms, proton motive force and chemiosmosis, they're really used to describe kind of the same thing that's going on there. But that potential energy outside of the cell, we describe that as the proton motive force. And then when they diffuse back in, that's the process of chemiosmosis. All right, that's when the actual generation of ATP takes place. So I don't know, that may help clarify those two concepts a little bit. I want to go back to this just for a second, just haven't talked about the final electron acceptor. For aerobic respiration, that's going to be oxygen. So oxygen is actually split, and one O combines with two electrons and gives us water. Now that doesn't always happen. The splitting of oxygen doesn't always happen just right. Sometimes the electrons are going to arrange themselves inappropriately, and we end up with superoxide. So that's O. Okay. And remember we talked about last time, that's one of those toxic oxygen forms. Okay, so when organisms respire aerobically, they get some superoxide just as a byproduct. So they have to have a way to detoxify that. They have to be able to make that enzyme superoxide dismutase. In addition, sometimes we mentioned last time how electrons have to be chaperoned around the cell wherever they go, because they'll cause damage if they're not kind of escorted wherever they need to go. Well, sometimes the electrons aren't passed correctly down the electron transport chain, and you get some electrons kind of leaking into the cell. And so that's another way that superoxide can be formed, just that leaking of electrons. Okay, and as well as peroxide anions. Okay. So things like superoxide dismutase, catalase, peroxidase, those are all enzymes that will help detoxify those toxic oxygen compounds. Okay, questions about so far? All right, so kind of to wrap up respiration, aerobic respiration. If we talk about per glucose, and in a minute we're going to talk about, well, what if it's not glucose? And then we'll talk about, well, what if it's some other electron acceptor other than oxygen? But this is, at least in theory, what we get from respiration, both bacteria and eukaryotes. All right, so we talked about in glycolysis, you put in 2 ATP, you get out 4, so you net 2 ATP here. 2 ATP Krebs cycle, substrate level phosphorylation, and then all of the electrons that go to the electron transport chain, we get about 3 ATP per NADH and estimated 2 ATP per FADH2. Okay, so that's how we get these numbers 30 and 4. Okay, so if we add this all up for prokaryotes, 34, 38, about 38 ATP in theory. For eukaryotes, they say 36 because the cell has to spend 2 ATP to bring, it would be this NADH to transport that into the mitochondria. Okay, so at least in theory, the yield is just a tad bit less for eukaryotes. Now, I emphasize theoretical because actually organisms will actually use that proton motive force sometimes for other things. So for instance, bacteria, they will use the proton motive force to power flagella movement. Okay, not so for eukaryotes, they use ATP. But so they don't theoretically, get this kind of yield from the electron transport chain because some of those protons kind of go towards something else. Sometimes they will use that proton motive force just to transport things in or out of the cell. So again, that's kind of a theoretical best case scenario addition there. All right, so what if, I said that bacteria anyway will use glucose or some kind of sugars as their first choice of metabolism, because it's just kind of the easiest thing for them to use sugars. But if sugars aren't available, they typically will be able to use proteins or lipids if those are available. So this actually matches up really nicely with what we're going to do in lab next week. it used to match up just right, but for some reason we got off a little bit. Remember that bacteria can't just phagocytize some big nutrient and bring it into their cell because they have that thick cell wall. Okay, so in this picture, they don't really say that, but remember bacteria have a cell wall. So if there's some nutrient that they need to use, they have to secrete enzymes outside of the cell to break it down, right? So one of the labs next week, we're testing for different extracellular enzymes that are secreted outside of the cell. Organisms have a variety of different ones, some they can make, some that they can't. But if they are secreting some enzyme with the goal of breaking down a protein, we would call it a protease. Okay, so in lab, you're going to test an enzyme that can break down the protein casein, call that caseinase, or a protein that can break down gelatin, call that gelatinase. Okay, and that splits some polypeptide, a protein is a polypeptide, down and into its constituent amino acids, all right, cleaves those bonds there. And then the amino acids are small enough that they can easily be transported into the cell. There's different ways that they can be metabolized, but often the first step is that amino group is removed. We call that a deamination. Right, so one of the enzymes, for instance, that we're testing for in lab, or another one, is phenylalanine deaminase. If the cell is going to be able to use phenylalanine as some kind of energy source, that's kind of the first step, remove that amino group. And when the amino group is removed, you're left with some 2-carbon compound here. And remember, we said 2-carbon compounds can get shunted right into the Krebs cycle. So now that compound can, we can get all the electrons, especially out of that, be converted into energy. And incidentally, this NH2 typically gets converted into NH3, also known as ammonia. And when you see ammonia, you think alkaline pH. So that's a big difference. When organisms metabolize proteins, they get end products that are alkaline. When they are either metabolizing sugar, no matter how they do it, it's almost always acidic. Respiration, only a little acidic. When we talk about fermentation, really acidic. So that's something, again, that we can test. What if a bacteria wants to break down lipids, a lipid? Once again, this is an extracellular enzyme that we're going to test in lab. This lipase, an enzyme that breaks down lipids. And lipids are made of fatty acids and this 3-carbon glycerol. Okay, so an enzyme that hydrolyzes that is going to cleave this glycerol off from the fatty acids. Okay, so kind of a brief look, again, in your biochemistry class, you'll pick this apart a little bit further. A brief look of what happens then to the glycerol and the fatty acids separately. That 3-carbon glycerol, a little ATP gets inputted there. So now we have this dihydroxyacetone phosphate. You may or may not recognize that when in the splitting step of glycolysis, I talked about how that molecule, it's not symmetric. It doesn't just get split into two, phosphophenol pyruvate or glyceraldehyde 3-phosphate. It's not even. You get one glyceraldehyde 3-phosphate and one dihydroxyacetone phosphate. That usually gets converted then into glyceraldehyde 3-phosphate, and it can enter in kind of the bottom half of glycolysis there. So get a little bit of ATP from that, and then lots more electrons in the Krebs cycle. But it's the fatty acids where you get just a ton of energy produced from that, because each fatty acid will undergo beta oxidation. Okay, it's called beta oxidation because this carbon, the second one from the end, That's the beta carbon. Okay, and what happens, that's cleaved off 2 carbon compounds here. That's the process of beta oxidation. So basically we get some reducing power here when that happens even. So we get the cleavage there, that coenzyme A gets involved, cleaves off with that 2 carbons, and that gives us acetyl-CoA, which you should recognize as being what we get after that transition step of glycolysis. That's what goes into the Krebs cycle. So that can happen, you know, over and over and over again with each fatty acid. So all those electrons again get converted into ATP at electron transport chains. So lots of ATP from that beta oxidation process. Okay. All right. So what happens if, remember, bacteria are often, they're a lot more flexible than we are. They can often use some other kind of electron acceptor other than oxygen. Okay. And it's almost always going to be some inorganic ion. I think there's a few exceptions to that, but usually some inorganic ion. So remember, that means there's no carbon to hydrogen bonds. Whoops, sorry. Don't put that double bond there, right? No carbon hydrogen bonds. So some kind of inorganic ion. So what are some examples of things that can be a final electron acceptor? Nitrates. Okay. If organisms have the enzyme nitrate reductase, so they can reduce nitrates to nitrite. So again, this is nitrate here. This one is nitrite. If they have that enzyme. And I talk about that in a little bit more detail because that's another enzyme that you're going to talk about next week in lab. Okay, if organisms can make that enzyme nitrate reductase, and I didn't post this, but that's an enzyme right here, this green one, that's going to be right there in the membranes of a plasma membrane. If the organism has that, enables it to use nitrate here as a final electron acceptor. All right, we don't make that. We are not able to do that. So a lot of bacteria can. This whole big group, the Anaerobic Bacteriosine, that's the E. coli is in that family of bacteria. So a lot of organisms can do that. Some organisms can use sulfates. If they do, they could get hydrogen sulfide as an end product. That's another gas that we're going to test for kind of near the end of the semester. It's that smelly gas, like if you've ever been to Yellowstone, that kind of rotten egg smell, that's hydrogen sulfide. So that's a kind of a byproduct of an organism respiring anaerobically. Sometimes iron is a final electron acceptor. Sometimes carbonate. Okay, and bacteria that use carbonate as a final electron acceptor, or sometimes carbon dioxide, can actually produce CH4, which is methane. Okay, this is actually archaea bacteria, archaea, that can do that, produce methane. So lots of these organisms in the rumens of cows, lots of these organisms. One other place that you find them is in the landfill. Okay, so what do they do in landfill? You've got lots of trash, lots of organic stuff that's being covered up by soil. A nice anaerobic environment gets packed in there, we can actually, and then that's where these methanogens grow and produce methane. So they actually have to vent landfills kind of carefully because methane is an explosive gas. And I tried to look that up, like how common is it for you to have like a fire or an explosion at a landfill? I guess it's not that common because we usually, hopefully do things correctly. But we can actually catch some of that methane that's produced by bacteria and use it to run the landfill or use it to produce electricity. It's not really economically feasible at this point. It costs so much to kind of, for the apparatus to collect that gas, that it's not done very often, but the landfill just south of Grand Rapids does that on a small scale, enough to, like, you know, heat their offices there, little electricity there. Okay. All right, so what point was I going to make here? Okay, so it's important to recognize, and sometimes we get mixed up between anaerobic respiration and fermentation. I don't think I posted this again, but again, if oxygen is the final electron acceptor here, that's aerobic respiration. If something other than oxygen, like say NO3, okay, that's anaerobic respiration. Okay, so if the organism's using something like nitrate, it's still going through glycolysis and having a Krebs cycle and having an electron transport chain. The Krebs cycle might be modified a little bit, but if it's using something other than oxygen, it's still respiration. Okay, so we're going to get to fermentation next in a second, but I want to make one more point about anaerobic respiration. And I could just tell you this, but I want to try to explain why this is the case as well. So that requires me to talk about this redox tower. The point that I want to make, and I'll just tell you this right off, that bacteria always are going to get less energy and I'm comparing it to aerobic respiration. They'll always get less energy from anaerobic respiration. Okay, so they can use other things besides oxygen as a final electron acceptor, but they're never going to get as much ATP out of it as if they had used oxygen. And the reason is, remember I said we can measure electron potentials. We stick these electrodes in cells. It's kind of astonishing that we can do this. But that's how we know actually the order of the electron carriers. So you recognize some of those here, cytochromes, ubiquitinone. You know what order they are because we can measure how badly they want to give up electrons or how badly they want to take electrons away. Okay, so we look at the redox potential, this E naught prime, that EO prime, that's pronounced E naught prime. That's a measure of the ability of some molecule to give up its electrons. So if that E naught prime is very negative, that means they want to give up electrons very badly. They want to get rid of their electrons. If that E naught prime is positive, the more positive it is, that means they want to take electrons. All right, so if we measure the distance between, for instance, what we're starting with at our electron transport chain, so this NAD-NADH pair, if we measure the distance between that and then the final electron acceptor, oxygen, okay, that's a pretty big distance here. Okay, so the bigger the distance, the more ATP is generated. So again, I'll leave that to your biochem professor to figure out that calculation to figure out how much energy that we get. But that's aerobic respiration. If you use anything else other than oxygen, that distance is not going to be as great. All right, so if you say, let's see, electrons dropped off by NADH, if you use sulfate as a final electron acceptor, okay, that's going to give you less ATP. If you use nitrates, a little bit more, still less than if you use oxygen. Some organisms can further reduce nitrite down even further, so they could get, that's a little bit lower here. Iron almost as far as oxygen, but not quite. So that's a little bit of why you're always going to get less energy. But of course, the trade-off is, that it enables bacteria to live in places without oxygen. Okay, so they might be able to live somewhere where there's a whole lot less competition. If they don't have to compete with aerobic organisms, you know. And this is, you know, swamps or, again, landfills, places that you're not going to have a lot of access to oxygen. Maybe even in our gut. Okay, there's probably a little anaerobic respiration going on there in our colons, for example. Okay, so who does anaerobic respiration? These are organisms that are considered facultative anaerobes. Remember, facultative, that means flexible metabolism, aerotolerant anaerobes, strict anaerobes. Pretty much everybody except strict aerobes. Now, the tricky part is, we're going to talk about fermentation, yet there's always a lot of confusion because some people would define fermentation as any metabolism that doesn't require oxygen. Okay, we're going to refine that definition a little bit and talk about, well, what's the difference between fermentation and anaerobic respiration? The difference is really what happens to pyruvates. Okay. That's a big branch point, and they're really totally different pathways. Fermentative pathways are totally different than anaerobic respiration pathways. Okay, so going back to our overview here, glycolysis is part of both respiration and fermentation. But then this pyruvic acid, this is a big branch point. If there's some final electron acceptor available, it's going to go a respiratory pathway because it's going to get more ATP that way. If there is not a suitable electron acceptor, the cell has no choice but to enter into some fermentative pathway after glycolysis. Okay, and the whole point of the fermentative pathway is to recycle this NADH back to its oxidized form. Because if you get a bunch of these NADHs building up coming off of glycolysis, they're all going to be saturated and there's no more NADs to keep these oxidation redox reactions going for even glycolysis. So if you don't get that NADH recycled back to NAD, even glycolysis will stop and don't get any ATP from anything then. Okay, so that's the big point of fermentation. Usually we're not getting, we'll say often, we get no additional ATP. Okay, that's not always the case. Sometimes there's a few more reactions past glycolysis that we get a little ATP, but sometimes not. Okay, and we always have a organic molecule that's the electron acceptor. So that means something that's already there in the cell is acting as an electron acceptor in order to accept these electrons, these two electrons that are coming with the NADH. Again, oxidize that back to NAD. Okay, so your book doesn't really talk about fermentation all that much. Here's the one figure. They talk about a couple of different fermentative pathways. There are a lot of different fermentative pathways out there. You take a good microbial physiology class and we'll go through a bunch of them. The most common one is when a cell takes pyruvic acid, accepts those electrons from NADH right here, and you end up with lactic acid. So here's the only difference here. We got a hydroxyl group here instead of this oxygen right here, pyruvic acid. Okay, so no other ATP, there's no gases given off. That's the simplest fermentative pathway. There are bacteria that what we call homolactive. That means all they do is lactic acid fermentation. They don't do any kind of respiration at all. They get all their ATP just from what they get out of glycolysis. Okay, so they burn through a whole lot of sugar getting all the ATP that they need. We can do a lactic acid fermentation in our muscle cells. We absolutely have to. If you've ever run a 400-meter race, the reason that you couldn't move your legs at the end is because there's lactic acid building up in your muscles. Okay, so we can do that on kind of a short-term basis in some of our cells. A lot of bacteria do it. Another pathway, so this figure makes it look like, oh, you can go either way. Well, no, some organisms can do that. A lot of bacteria can. Other organisms like yeast will do an alcohol fermentation. Okay, so you end up with some end product. There's a lot of kind of energy left in that end product, but that's pretty much without oxygen. That's pretty much all the energy that the cell can get out of it. Again, recycling that NADH. Here we get a little carbon dioxide. It's very common to have some kind of gas as a byproduct of a fermentative pathway, but not always. Okay. What else did I want to say about that? So here's an example. I didn't want to post this because I didn't want to scare any. Before I get to that. So this week in lab, and I lots of ties into lab this week. Again, for your benefit, if you're not in lab, and also for your benefit if you are, to tie it in. We're going to be testing for fermentation in lab Wednesday and Thursday. So you're going to have these tubes. We either call them fermentation tubes, or sometimes some of you call them purple tubes. They're purple, and each tube has a different sugar in it. So one will have glucose, one will have lactose, sucrose, mannitol. We're going to inoculate our organism and see if they can ferment that sugar. Sometimes organisms will be deficient in an enzyme, say, that might split a disaccharide, and then they can't utilize it. So we want to see if they can utilize it. If they can, they'll have lots and lots of acidic end products, like pH 5, like pretty acidic. So we look for a color change, all right? Yellow color change, got a pH indicator in there. Yep, it's fermenting that sugar. We also look to see if the cell is making some gas as a byproduct, because we actually have another little tube inside of here. If you look close, there's a little gas bubble at the end of that. Okay, so that tells us, oh, that organism is using some pathway that it produces gas. And again, that's not always the case. For instance, this organism, you can see this little tube in there. There's no gas bubble in there. So we will be testing for fermentation this week in lab. There are lots and lots of other fermentation pathways. So I didn't post this because it looks a little bit wild and crazy. But this is a good example of, bacteria can do some really nasty stuff. They can also do some things that really help us. So for example, again, using pyruvate as that final electron acceptor, there are some organisms, these propionibacteria, that will produce propionates as a byproduct. That is the distinctive flavor of Swiss cheese. Okay, so bacteria, they're secreting carbon dioxide as a byproduct there. That's the pools that we get from Swiss cheese. So thank you, bacteria, for that. There are some pathways. There are a few other pathways that you're going to test for if you're in the lab later in the semester. There's a 2,3-butane diol pathway. This is actually what we call mixed acid fermentation pathway. that helps us identify organisms if we can understand what pathway they're able to do. The Clostridia, these are some organisms that we've used in lab last week and this week. They can do some really interesting things. This butyrate intermediate, that is supposed to be the smell of C. diff enterocolitis. So the whiff of the diff, it's supposed to be like the nastiest smelling thing that you can encounter. It's supposed to be pretty bad. And it's because they're doing this particular kind of fermentation. Okay. So bacteria can be pretty smelly. It's usually because of fermentation. So the whole deodorant industry is basically, you know, has bacterial fermentations to thank for that. Bacteria are multiplying on warm, moist places in our body. So again, bacteria do some great things for us because of the fermentations that they can do. I mentioned the Swiss cheese, organisms that do this lactic acid fermentation, like Streptococcus, that may be one that you've heard of, you'll have strep throat probably. That helps us make cheddar cheese, yogurt, soy sauce, of course, yeast, all of our alcoholic fermentations, Clostridium acetone, isopropanol, things like nail polish remover. Okay, that's actually comes from bacterial fermentation. Acetic acid, of course, is vinegar. So a lot of bacteria that make that. So not to memorize those things, just to kind of give you a little appreciation, maybe. And to kind of wrap up, there's a table there in your book. It's one of those tables that's good to make sure you're getting the big picture. Like, you know, cover up aerobic respiration and see if you can kind of answer those questions. So that's kind of a nice little good sum up table in your book. Okay. A lot of questions that I get asked is, you know, what do I have to know for metabolism? So here's a couple examples of questions that I've asked before on tests. So I like to ask, okay, what pathway does this, or what pathway does that? I'll give you a second to think about that. Okay, so remember we said when pathways use NADP as a electron carrier, that phosphate tags it to go towards a biosynthetic reaction. So the one that uses that on this list is pentose phosphate. Okay. Also get a little NADP from the Entner-Doderoff, but that wasn't one of your possible actions there. Remember, this is the same thing as glycolysis. And one more. Okay, again, a lot of sometimes confusion between anaerobic respiration and fermentation. So which one of these is true? All right, hopefully you chose B, since we just talked about that. All right, so these are all describing fermentation. Of course, sometimes fermentation does give you a little ATP. Anaerobic respiration will give a lot of ATP. It's not quite as much as aerobic respiration. Question?
So with fermentation, our bodies, do we use it when we don't have access to oxygen? Is that the reason that it gets triggered?
So, right, the question is, do we use fermentation when we don't have access to enough oxygen? Right, right. So in our muscle cells, when we're working hard for a long period of time, that's when, that's my understanding, that's when they'll utilize fermentation, right? Yeah. Okay, well, let's forge on Okay. Remember, so you can be working on that chapter 5 homework. Remember, I would skip to this if it was easy for me to navigate to it. But remember on mastering, there is a whole section, there's a tab that says study skills. Okay, and then you can navigate to whatever chapter you're working on. Say for chapter 7, we are doing most of chapter 7. We did most of chapter 5 too, although we skipped photosynthesis. But in there, there's going to be, there's a post test, there's a pre-test, there's some extra questions there that you can practice with. Again, skip the photosynthesis questions because we didn't talk about that in here. Chapter 7, we are doing most of chapter 7. I actually haven't posted quite all the PowerPoints. I haven't posted the mutation stuff yet. So it's a pretty long chapter. The homework has a little bit more animation type things, which is pretty good. All right, so take advantage of that. You can actually go in again to the study part and access even more animations if you see something that has been confusing to you and get some extra work on it. Okay, so first of all, structure of DNA. So hopefully a little bit of a review to start with here, and how that DNA, kind of an overall, how that gets replicated. That's a famous experiment we're going to look at there. And then we're going to get to the nitty gritty of how replication takes place. That's the copying of DNA, how that DNA gets made into RNA, call that transcription, and how that RNA gets changed into proteins, call that translation. All right, so we're actually going to bring in a few figures, not from your book. I think the book's covering of transcription in particular is kind of wanting. So I'll bring in a few other things, hopefully that will clarify instead of confuse you. It's important that cells aren't making proteins that they don't need, because it's a very energetically expensive thing. So we'll talk about regulation for bacteria that involves operons. And then a little bit about mutation at the end and how cells can fix that, as well as how bacteria can share DNA. So we call that horizontal gene transfer. All right, so kind of a long chapter. Use your book. Probably, I would say use that more of a reference. So, you know, transcription, translation, it's kind of confusing. What does the book have to say? Hopefully it can clarify some things. Okay, so structure of DNA. This shouldn't be new. We got deoxyribose. These are the pentose sugars in what we say the backbone. Phosphates in between those sugars, and there are nitrogenous rich bases that are bonded to that sugar. Okay, and of course bonded to their complementary base pair on the other strand. So double-stranded DNA. The adenine and the guanine considered purines. I remember that because I went to an ag school, and I always say Aggies are pure. I don't know, that's what helps me remember adenine, guanine, they're purines. And then cytosine and thymine, they're the pyrimidines. Okay, and of course the A's always bind the T's with 2 hydrogen bonds. And the G's always bond to the C's. You can see that there's 3 hydrogen bonds. Okay, so that the number of bonds that kind of plays into, we see that anytime that any kind of copying of DNA is happening, where it begins, there's usually an AT-rich region because those are kind of unzipped a little bit easier. Sometimes, you know, when it's terminated, you have a stretch of A's and T's. So again, that's a breaking apart of those bonds can happen a little bit easier. Then your book goes into a little bit more detail here. This is helpful just to kind of orient us. Of course, DNA, double-stranded DNA, that was elucidated, that structure, by Watson and Crick in a very famous one-page paper, I think 1953. They didn't do any experiments to figure out the structure. They just kind of synthesized all the other information was known at the time for that very famous paper. It's anti-parallel. So that means the 3' end matches on one strand matches up with the 5' strand on the other. Okay, we call that anti-parallel. Whoops, that's supposed to be an L. So what does 3' and 5' mean? Remember, if you look at the deoxyribose sugar, it's a pentose sugar, but there's 5 carbons. They're not all in the ring though, and we number them. The first carbon, or 1' carbon, is the one that's attached to the base. And then we count from there, 2', 3', 4', and then the 5' carbon is the one that kind of sticks up that's not in the ring. All right, so that means this is the 5' end, and where this 3', there's actually a hydroxyl group here that bonds to the phosphate. Okay, that's the 3' direction. Okay, and then the other strand is oriented the other way. Okay, so this is the 1, the 2, the 3, the 4, and the 5'. All right, so here's the 3' end of this strand and the 5' end of this strand. All right, so we will talk about directionality a little bit and try to keep that straight. The other terminology here that I'll use, a nucleoside, that's the base and the sugar. So like a thymine nucleoside is thymine base and deoxyribose. A thymine nucleotide will also include the phosphates. Okay, so nucleoside without the phosphate, nucleotide includes the phosphate. That terminology can get a little bit cumbersome. Okay. So structure of genomes, we talked about this kind of real briefly when we were talking about structure of cells. Of course, where our focus here is on prokaryotes, but we'll compare that to eukaryotes. In terms of chromosomes, that's where the genes are. Prokaryotes are usually going to only have 1 chromosome. It's usually circular. And because when it's circular, the ends are not going to be exposed. Okay, so our chromosomes are linear, got these exposed ends that are kind of open to being degraded. So it's kind of a nice stable form for the bacterial chromosome to be. They want it that way because it's always going to be haploid. Okay, that means that they have 1 copy of each gene. You've only got 1 shot at it, so they gotta protect that. And remember, that's going to be found in the nucleoid region of the cytoplasm. So it's kind of hard to see from this picture, but you know, remember, it's just not enclosed on membrane. Compare that with eukaryotic chromosomes. Of course, they usually are going to have multiple chromosomes. Right. We have 23 pairs of them, so we have 46 chromosomes ourselves. They're going to be linear and diploid. Okay, we have 23 pairs. We have 2 copies of each chromosome. And they're going to be found in the nucleus. Remember, the one exception to that, the DNA of our mitochondria and chloroplasts is circular. So there is some DNA in our cells, technically, that's more like prokaryotic form. Now, in addition, prokaryotes, bacteria and archaea, will also have plasmids. And these are small, much smaller circular molecules of DNA. They don't replicate on the same timetable as the chromosomes. So they can kind of replicate whenever they want. And typically they will carry instructions for proteins that are not absolutely essential for survival. So for instance, any enzyme that is necessary for glycolysis, that's going to be on the chromosome. Maybe an enzyme that helps an organism survive in the presence of antibiotics, gives it just a little advantage, that's going to be on a plasmid. So we have different kinds of plasmids that a bacteria can have. A fertility plasmid, for example. We talked about pili. We talked about pili, a conjugation pili. We'll talk more about what conjugation is next week at some point. But if the organism has this plasmid, that fertility plasmid that enables them to make a pilus, a resistance plasmid carries instructions to help them survive in the presence of some antibiotic. Talk more about bacterial resistance in a little while, 2-3 weeks from now, probably right after spring break. There are these plasmids called bacteriosine plasmids, and they carry instructions for a cell to make bacteriosins, which sometimes some scientists would call bacteriosins a very, very specific antibiotic. Basically, they're chemicals that a bacteria can secrete to kill its own species or a species that's very similar, okay, if they don't have the plasmid as well. And then another big group of plasmids, virulence plasmids. That's the instructions for any kind of gene that would help a cell be more successful in colonizing a host. So that might be some anti-phagocytic mechanism. It might be a toxin, for instance. We're going to talk much more about virulence factors going forward. Okay, so I think we just have enough time to talk about DNA replication in general a little bit. We'll end with this. But when Watson and Crick wrote that paper about the structure of DNA, they didn't really have they weren't sure how the replication of the DNA took place. There was really 3 hypotheses at the time. They said it could happen semi-conservatively. So that means one of the strands of this original strand was conserved in each, strand that was copied. Okay, we call that semi-conservative. The conservative hypothesis was that this original strand was unwound and copied, and those 2 new strands were totally new. And then the old DNA was conserved. And then there was this other hypothesis that there are enzymes that go through and chop it up like this, and you get kind of an old stretch and a new stretch and an old stretch again. Okay, so those are the 3 different hypotheses. There's a very famous experiment that I want to talk about called the Meselson and Stahl experiment. And this has been called the most beautiful experiment in biology because not only did it prove which of those methods was correct, but it also disproved the other 2. There's a very interesting YouTube video that I stumbled on, an interview with Meselson and Stahl. Okay, so this experiment that this short clip is going to talk about, they took place in 1958. So these 2 guys, Meselson and Stahl, they were young men when they did this experiment. They were given kind of freedom to do whatever they want. And there's an interview with them on YouTube that was just from a couple of years ago. And they're really hilarious. Like they're old men talking about, you know, them doing this experiment. So this is just a 2 minutes clip. And I'm probably not going to have sound, come to think of it. But this YouTube clip explains it better than I could. I could attempt and struggle through that. Why is that? I don't know why that's sticking out like that. But this again, it's just a 2 minute clip. They used a heavy form. Maybe I can try to talk you through it.
The Meselson and Stahl experiment provides evidence for semiconductor replication of the DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Meselson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
Okay, so
the Meselson and Stahl experiment provides evidence for semi-conservative.
So basically, you know, if you're printing in black and white, this isn't going to make a lot of sense. But this talks about the different generations and where the bands would be. And I included this figure as well as to this is the pattern that we found. But if dispersive was true, you know, we'd have these intermediate bands and never just a heavy and a light. Okay, so it's just, again, most beautiful experiment in biology because it was very effective at disproving. And those two guys in that interview, they said they presented their work at a conference, and somebody in the front row said, "Well, I guess that's it then." You know, wouldn't we love to have an experiment that would prove climate change is taking place or proving it's not? You know, there's always so much, you know, a huge population of people that don't believe this or that. So it's nice to have an experiment that kind of sums it up really nicely. All right, so next time we will jump in with sort of the molecular step-by-steps of replication and get into transcription and maybe even translation. So see you next time.